So I'm going to talk a little bit about, well, first, thanks for having me back. It's always nice to be invited back. Maybe that means that you did something good the first time. So I will catch you up on what I said last year, and then really spend most of the time on this Meth2 transcriptional activator. And I just want to give a shout out to Matt, because I was able to remove half my slides because he gave a lot of the IPS and CRISPR-Cas9 that I was going to talk about. So that's fantastic. I can just show you the results without having to worry about that. Matt actually mentioned this, that excitatory-inhibitory imbalance is thought to underlie many of the abnormal phenotypes in ASD. I'm having an issue with my screen here, but I think I fixed it. The transcription factor Meth2c is involved in multiple forms of autism and intellectual disability. And so we hypothesized that boosting its activity might normalize EI imbalance and neural behavioral abnormalities. And we screened what's called the Scripps Reframe Library. The Scripps Reframe Library is a unique library that Scripps chemists, Scripps, I am so honored. Scripps has, it's been ranked 1 or 2 in the world in chemistry. The chemists think I know some chemistry, so we're all going to be quiet about that. And they put together a library of not only all the FDA-approved drugs, as Matt had mentioned, but all the EMA-approved drugs in Europe and other compounds all over the world that have made it through phase 1 safety trials, even if they're not yet on the market. So the beauty of that approach is that we have all the drugs basically that have been ever tested in human that are safe, and we are able to test those. And the lead candidate I'll show you today has extensive experience in humans for another indication and shows excellent Meth2 activation and corrects EI imbalance. And I'll show you some of that data. Okay, so this is bringing coals to Newcastle. You've heard this in multiple other talks. The latest CDC statistics are now 1 in 44 children. You've also heard Dan Geschwin's name in both Sarkis's talk and Joe's talk. There are multiple slides. Dan is also my colleague, and he looked at, I'll show you the data in a moment, a series of ASD genes and was able to parse those genes into families. And interestingly, the transcription factor Meth2c and Meth2a, a related transcription factor, control many of those genes. Dan then contacted me. I was the discoverer of Meth2c, and with Dan, we've been looking at Meth2c in various neurologic diseases, including autism spectrum disorder with intellectual disability. So our reasoning is that therapies aimed at improving Meth2c-associated ASD phenotypes might potentially affect many other forms of ASD, because we know, not from iPSC data, although we do know, from real human data, that Meth2 controls many of the genes involved in ASD and intellectual disability. So here's some of Dan's data from a paper in Cell, and you can see these 5 hubs of genes that are many of the genes that Joseph showed you in the prior talk. And Meth2c and Meth2a are 2 of a few transcription factors. Transcription factors are proteins that go in and turn on genes. So they control many of the other known autism genes. Now, interestingly, Meth2c itself can be mutated and form a very severe form of autism and intellectual disability. We first discovered this actually in a mouse, and then colleagues subsequently discovered it in humans. And so this is a gene that encompasses the Meth2c region, and now we know specifically of many mutations in Meth2c that cause this. If you look at the UK database and some other databases, it has the possibility of being extremely frequent, in that more frequent than, say, Rett syndrome. Oops. And these children generally have severe autistic behavior. Only a few of them talk, accompanied by epilepsy, periodic tremor, and abnormal EEG. We'll get back to the electrical activity in a minute, and often severe intellectual disability. Although there are a few that are less severely affected that I've seen. I'm also a physician and have seen, it must be nearly 100 of these cases now, and I'm in contact with many of the families. So we have 2 disease models. One you just heard about. We also use human patient skin biopsies, both from patients that we then use CRISPR-Cas9. We take tractable mutations. These are mutations that are not too large that we then can correct. We've also introduced mutations into the normal genome to mimic Meth2c. So we have a whole family of these Meth2c mutants. Importantly, we've used multiple genetic backgrounds. You must do this to ensure that it's not something in the background that's causing your phenotype. So we have them from multiple families that we've corrected. We also have a Meth2c mouse that we published, shown here, and we use that to test our drugs. Although, like Matt Lally, we feel it's really important to do this in a human context in iPS cells. Okay, so let me show you our disease in a dish model of the human Meth2c haploinsufficiency syndrome. And so, as Matt just eloquently explained, so I was able to take out many of my slides. We take patients, make human iPSCs, then we can differentiate them into neuroprogenitor cells, into neurons, and multiple other cell types. We've done that not only in two-dimensional cultures, but in these 3D mini brains called cerebral organoids. I'll show you some pictures in a moment. And this just takes you through the stages. These show that we've made NPCs, that is pluripotent cells. We then can take them through the various stages to make neurons. They start to look like neurons, but importantly, they stain to show their neurons. They have the electrophysiological characteristics of neurons. We also have done single-cell RNA-seq, and I'll tell you about that in a minute. So I think the most important finding from this, which should be appearing shortly in Science Translational Medicine, is that the Meth2c ASDs make a lot more astrocytic cells and fewer neurons. So here MAP2 is a marker for neurons, and you can see there's not a lot of green here in the Meth2c child's neurons. In wild type, estrogenic wild type, a lot of neurons. And the opposite is true if you look here. GFAP and S100 beta in this context are astrocytes, and you can see more in the Mef2c neurons after 6 or 10 weeks in culture, and fewer in the wild type. So you're not making neurons properly, you're also not making synapses properly. Yeah, so I mentioned we can make these 3D cerebral organoids in a dish. We've actually brought some of these kids in, told them we created a little piece of their brain or part of their brain in a dish. They look like this. Many of them actually have little ventricles, what you have in your brain, as you can see here. They have layering. I won't go through all the different layering we have, but we can get them pretty mature. We've kept them for 6 or 9 months. We do this in multiple diseases, Alzheimer's disease, which I'll come back to at the end, actually. But I'll show you the autism studies today. And then single-cell RNA-seq shows multiple cell types, interesting deficits in both excitatory and inhibitory neurons in the Mef2c ASD patient samples versus wild type. And others have shown that Mef2c is very important in both excitatory neurons and inhibitory neurons. Interesting also in human microglia, although I won't talk about that today. But we think that underlies some of the infections these kids get. Okay, so let's cut to the quick. What do these cerebral organoids do? Well, they manifest EI imbalance, excitatory inhibitory imbalance. They actually have too much excitation, and this is one indication. We have patch clamp recordings. This is calcium imaging. We have multi-electrode arrays where we can record the whole brain like an EEG. I just want to show you calcium imaging. I showed some of this last year, but we have a lot more to show you now. And so you can see this is the normal wild type, and the neurons are signaling to one another. And we can quantify this. I'll show you the quantification in a minute. If you look at a kid, a child, iPS cells, this child happens to have seizures, and whoops, we've actually recreated the seizure in a dish. You can see it pulsating like that. We can quantify that. Not all of them have seizures. Sometimes they just have too much electrical activity. So the EI is balanced in this case toward E. Too much excitation, you can see it here. Last year, I talked about a drug that we're developing called Nitrosynapsin, and you can see when we put it on, it calmed things down dramatically. Here it is again, when we calm it down, see everything gets much more toward normal. So as I mentioned last year, this is an interesting type of glutamate receptor antagonist called the NMDA receptor. It's based on a drug that I patented actually for Alzheimer's disease. It's now one of the FDA-approved drugs for Alzheimer's disease called memantine. We modified the structure of memantine, and it has a second site of action at a site we call the S-nitrosylation site, more chemistry that had been discovered in my lab some years ago. So this is a dual-acting, very, very potent drug, but very, very safe. Little company in Boston is developing it. Here's an update on that, and then I'll get to the Meth2 activators. So I won't show you again, but I showed you last year, and we published it, that this drug, nitrosynapsin, damps down only the excessive excitation, doesn't affect normal excitation in the brain. It also corrects multiple neurobehavioral abnormalities in our Meth2c mice. We've now nearly completed pre-IND studies in preparation for a human clinical trial. We're actively seeking financial support. There's a major philanthropist who's involved in autism who's considering funding the trial, but we'd love to include any of you if you're interested. We are actively raising funds for the trial. We're going to do a phase 1/2 trial at the same time. And we've identified the first site, which will be at Mass General. They've developed a clinical biomarker that reflects this excessive NMDA receptor activity, so we can get an early readout with our biomarker in addition to doing neurobehavioral tests. So that's the plan for nitrosynapsin. Let me now turn to this Meth2c approach, which we're very excited about, and which the Brain Foundation and the Meth2c Association have been funding recently in the lab. And so I mentioned that obviously Meth2c haploinsufficiency, where one copy of the gene is missing or has a deletion or is somehow not working, there's one allele, that the other copy of Meth2c that's still there. Perhaps we could overstimulate that and compensate. And then most importantly, because Dan Geschman had shown that Mef2a and C are very important in controlling those hubs of many, many other genes involved in ASD, we reasoned if we could increase Mef2 activity in general, we might be able to turn on those genes and offer some benefit. And so we carried out high throughput screening of the chemical libraries I mentioned at Scripps, not only that Reframe library, but multiple other libraries. We have about 1.5 million compounds. I haven't screened all of them. I don't believe in that mindless screening. We screen for drugs we think are in categories that can work. So we have about 100 case screen that we've done. And we've identified 5 drugs that are existing drugs, already been in humans. And we have one candidate, I'm going to call it compound Y today, and I'll show you some data with that compound. So how do we do this? Well, as I mentioned, Mef2c and A, and there's other forms of Mef2, but those are the 2 that we know are involved here. They bind to a piece of DNA and turn on various target genes. What we've done is replace one of those target genes with luciferase reporter, something that bioluminescently has bioluminescence, so we can see it in the screening method. And then we add multiple drugs, and we see if it activates Mef2. And here it's shown in a 384 well plate. We have a luminometer to read that bioluminescence. We can put one drug in each, and we get basically certain drugs turn on Mef2 activity, shown here on the y-axis. We have what's called a very good z-score. We have a good, anything over 0.5 means you can go and screen your libraries, which we've done. So here's one typical screen of many that we've performed. And this is Mef2 activity. Remember, turning on that luciferase. So we're going to turn on all those genes that are turned off, not only in Mef2c haploinsufficiency, but in many other forms of autism. And you can see the top hit here is this compound Y. We do have other hits, but we're particularly interested in this one because it has very extensive use in people for another indication. And so now we more precisely look at Mef2c activity for the aficionados out there. We compare it to Ranilla. So we just want to make sure it's not turning all transcription on, but just Mef2c. Ranilla is to monitor background activity, if you will. And we have very good activity at Mef2 sites. So we would turn on Mef2a and C in point of fact. Now we wanted an indication. Remember I said the neurons weren't growing properly and they weren't growing out their neurites and their synapses. So we have multiple ways. I'm just going to show you one of them. We can actually look at that neuronal marker MAP2, which is all over the neurites, and we can quantify that as shown on this plot here. And what you see is there's more MAP2 when we treat the haploinsufficient neuroprogenitor cells that have less Mef2c. And you can see much more MAP2 protein. I won't show you pictures, but also we get many more neurites and synapses when we put the Mef2 activator back into the dish or into a cerebral organoid. Okay, so remember I showed you this picture of a child with seizures, boom, boom, boom. We can actually quantify that aberrant electrical activity, giving you EI imbalance. and that's kind of shown here. So wild type, you do have normal electrical activity. Here we're just looking at those calcium bursts. And you can see in the Meth2 patients, this is patient one, but as I said, we have half a dozen patients, you get these bursts of calcium activity. Whoops. And when we put on compound Y, we still get the normal activity, no more bursts, and it's quantified here. So we are able to rectify EI imbalance. We're now testing this compound to look at various behavioral phenotypes. So we're really excited about it because it's been in patients. It's interesting, it's not FDA approved. So that also gives us an intellectual property approach to it, so we can get some company interested in bringing it forward. So in summary, what I told you about today, and I was able to go quickly because Matt, fantastic job, Matt, in kind of giving a lot of the background. I could skip those slides. So we were looking at Meth2 haploinsufficiency syndrome, and particularly we're interested in this excitatory and inhibitory imbalance as a biomarker, if you will, of drugs that might improve not only Meth2c haploinsufficiency syndrome, but other forms of autism that have EI imbalance. Now, just amazingly, my close colleague at MIT, Li Wei Tsai, published in Science Translational Medicine last month that Meth2c is also important in various forms of dementia. We had been worried if we played with Meth2c later on after initial development, we actually might do damage rather than help. And I think that's a real worry about genetically interacting with any of these genes that Joe or Matt or I have been talking about. But here we have Li Wei, who's vetted this for us, and she's actually found in the oldest old people who have a resilient Alzheimer's or other forms of dementia, they have more Meth2 activity. And in a mouse model, if you decrease Meth2 activity, you get a form of dementia. Multiple mouse models of dementia have left Meth2c activity. And if you genetically increase Meth2c activity, you can abate the dementia phenotypes, memory, cognition, and multiple neurobehavioral tests that Li Wei and her group did. So we're now testing our drug in Li Wei's dementia models as well. And so what does this mean? Well, we think this EI imbalance, and we recently published a review on this in Annals of Annual Review of Pharmacology, is that at the extremes of life, life is a bell curve. We're all somewhere on that bell curve. At the extremes of life, your nervous system is very, very susceptible to EI imbalance. And it's not just the neurons. As other speakers have mentioned, other cell types, both the myelin-forming cells, the astrocytes, microglia, they're all involved in this EI imbalance in an intricate network. We know that, so is the extracellular matrix. And we think you're vulnerable, and you paradoxically at the extremes of life can develop this EI imbalance. And some of the drugs we use in one disease, say in Alzheimer's, might be amenable to being used in autism to correct the biological pathway that's being disrupted. And I showed you that the Meth2 activator compound Y increases not only Meth2c transcriptional activity, but this biomarker phenotype of EI imbalance. This is a drug that's been in people, has no significant toxicity, and so we're really excited about bringing it forward. So I've had tremendous help. Here are the people that have helped me, other labs as well. Here's Dan Geschwin, Jeff Rosenfeld here at UCSD, Nick Short, my bioinformaticist. I particularly want to acknowledge and thank Pramila at the Brain Foundation, who had the faith in us doing this at a very early stage of the work. Thank you. And Sarkis and Joe, whoever votes for this, thank you all very, very much. And also I want to acknowledge the Meth2c Association in Europe that I mentioned, which is a fantastic group of families with a tough disease, with Meth2c haploinsufficiency, who've banded together really all over the world to try to help with this kind of work. So this is where the work is done. It's a beautiful building right opposite the ocean. I better not talk about this. I know the weather back east isn't good right now. And you're all welcome to visit after COVID, please. And I'm glad to take any questions. Thank you.
Thank you, Stuart. That was really enlightening, and I would encourage people to ask their questions in the Q&A. There's one question, maybe I'll start, and Matt, feel free to jump in as well, but it's more of a general question on the iPSCs and how well they can model the diversity of neurons found in the brain, human, animal, what have you. I guess that's the first part of it. And secondly, to what degree does this correlation or lack of correlation with the diverse neurons, whether it's at the expression level, the activity level, the neurotransmitter level, to what degree does an intervention like a drug or a CRISPR intervention inform us about what neuronal subpopulations may be relevant in the human brain?
Yeah, so let me start with that, and then we can get Matt to chime in too, if you don't mind. That'd be really good. Yeah, you're right. Look, we have a developmental model in a dish. Luckily, this is a developmental disease, so it's not that we're trying to model a very old disease. Now, my colleague, Arnie Kriegstein, who has worked on development in iPS cells for a number of years, has wrote a series of papers and also editorials, if you will, about this question. Can you model in a dish a human disease? I mean, you're in a dish. So there's several issues here. One is the cells don't develop totally normally, even in the brain organoid model. They develop more slowly. And, I think particularly this is important for neurodegenerative diseases. You're 65 or 70 years old, not like autism when you get a disease. But interestingly, in the dish, they're under a certain amount of stress. And that seems to speed up the development. So even Arnold has agreed with us that we can mimic many of the features of a disease. But this is why having a biomarker of the disease that mimics the phenotype, like EI imbalance, is so critical here. What I'm telling you is I can, in a dish, mimic a finding that I found in mice and others have found in EEG in patients. And I can use that biomarker to use my drug discovery. So like Matt, we're also doing something called CMAPS Link 1000, where we have thousands of drugs that fix transcriptional activities, and we can screen drugs that way. I think that's fine. Matt, I actually didn't know that it had also been done in human neural progenitor cells. I may write to you to get some more data on that, because the database I have is not just for human neural progenitor cells. Now, let me address the single cell RNA-seq data. We do see a diverse number of different cells in our cerebral organoids. Okay, now I can't tell you we have every type of neuron, but we have looked against adult databases, and that's actually how we classify the cells. We look at a transcriptome signature, if you will, of, say, an excitatory neuron in the striatum. And we can match those, and we do see a vast repertoire of cells, but I know we don't see all the cells in the cerebral organoid, but we do see many of them. And so we're encouraged that because we have that phenotype, that electrical phenotype that we're very good at monitoring, we think we have some concordance, if you will, on the disease.
Sure. Thanks, sir. A couple more quick questions, I think. Is a planned trial for compound Y in patients with Meth2c mutations or in a different subset of ASD?
Yeah, so that's great. There is no, although we're working with colleagues to get this, we really don't have what they have for Rett syndrome, which is a natural history study of Meth2c, which would really facilitate our doing a clinical trial. While we'd like to do that, I think all of these drugs, and we've met with the FDA and discussed this ad nauseum, we're probably going to be doing it in another subset of ASD patients that are a little more tractable. Yeah, so both for nitrosynapsin and the Meth2 activator drugs. That's the plan right now, simply because the FDA is driving us that way, but we hope in the future we absolutely want to treat those children. Look, all of our children need help with this, and we're working hard on that, as we all are at the symposium.
Yeah, I could imagine having a pre-screen based on a narrow genetic subpopulation, which is make recruitment so challenging that it would be very difficult to even run the trial, right? I'm just wondering, maybe just dovetailing off that question, because Meth2c controls so many other genes, are there other either genetic or maybe physiological parameters one can use to enrich for a population that may respond to the drug?
There is, as I mentioned, the Mass General group has developed a biomarker that they'll be publishing and talking about soon, and it actually reflects the phenotype I showed you. And so we're very, very excited about that biomarker, because when you have a little company taking a drug forward, and I've done this several times now with memantine and other drugs, you have to kill a drug. You have to, this is the trick of a little company. You don't make a drug, you kill a drug, so you get to the right drug eventually. So having a biomarker for an early readout in a study is absolutely critical if it's a reliable biomarker. And we think we have one.
Joseph, you have a question?
Yeah, I was just wondering about, some of at least the nitrosynapsin and even this new compound Y, they seem to be working on, as you said, excess electrical activity. What about trying it in just epilepsies? Intractable epilepsies, for example.
Yeah, well, that will be one of the readouts. Many of these kids have epilepsy. So that is one of the endpoints we've discussed with the FDA.
Thank you.
Stuart,
Great talk.
One more question in the Q&A. Does nitrosynapsin activate mTOR?
Well, downstream it may. What it does is, I haven't looked at that specifically, but we know that it, well, we partly have looked at that. So we've tested it in a tuberous sclerosis model, and it's published. And I'm not going to be able to conjure up that immunoblot in my head right now. We've looked at the pathway, and it corrects the pathway. It does interact with the mTOR pathway. So the answer is yes. And we think that's because it's, in part, many of these genes are neuronal activated genes. And we know that Meth2 damps down this excessive electrical activity. Interestingly, it spares normal synaptic activity. That's why it and memantine that I developed previously are so well tolerated, as opposed to many other drugs that have tried to interact with the NMDA receptor.
Sure. And Joseph, is that a legacy hand that's raised, or do you have another question?
That was great. Sorry, I don't remember that one.
We'll move on. Thank you so much, Stuart.
Thank you all. Thanks for having me.
Absolutely. To the next talk by Kazu Takahashi. She's an assistant professor at the Department of Radiology at the Gordon Center for Medical.
Okay, can you hear me? Hello?
Yes, we can.
Oh,
Yes.
okay. Okay, so first of all, thank you so much for having me and bring me here to give to share my story about Manos binding lectin MBL. So I wasn't sure what to do, but so I decided to give kind of broad sort of background what MBL and my thinking about MBL at this stage. So at this point, MBL to me, it's multifunctional. I hope I can convince you what I'm telling you is true. And MBL is like sentinel, so providing constant surveillance through, and also kind of house cleaning, and also through coagulation. I'd like to call it bionic glue. And lastly, it is a disease modifier. So as long as you don't have anything, you may not feel it, or you wouldn't know till you look for it. But if you have underlying problem or some infection, then MBL deficiency in particular would make you feel something wrong. Now, I'd like to start with a big picture, at least to me. So thanks to host defense system, we have innate immunity, which kicks in second or minutes really after our body is exposed to danger, could be just injury or invading pathogen, and provide local containment. And one of this system is called the pattern recognition molecule, which MBL belongs to. And if we now this sorry, oops, sorry, so oops, sorry, I'm messing about. Okay. Now, innate immunity has linked to acquired immunity. Now, which takes at least a week, a long time, but provide systemic and target-specific protection, often quite for lifetime, since it has memory component in it. Now, what is MBL? It belongs to the collectin family, and as I mentioned, pattern recognition molecule of innate immunity. By itself, functions as opsonin, which facilitate phagocytosis by so-called professional phagocytes like a neutrophil and macrophage. And also in cells, had served as a post-translational glycosylation, so located at border of ER and Golgi, which was actually done by the other research group. And also MBL in complexes with a mass, MBL associated stem proteases activate the lectin complement pathway and also function like thrombin. Now, I mentioned innate immunity target is broad. So when we started MBL target, we really thought only pathogen. So bacteria, fungus, and virus. But we now know MBL can recognize much broader sugars, and ourselves, like once it's injured or transformed like a cancer, MBL can recognize also some enzymes and amyloid beta, like a Alzheimer protein. Now, as a function, by itself can serve as opsony, so increase phagocytosis by neutrophils and macrophages of MBL target. And in a complex with MASP1, MASP3, so MBL's functional MBL is hexamer. So then this complex activates the lectin pathway. So halfway through alternate pathway as well as classical pathway. Now, our contribution to this was this MASP1 MBL complex can function like thrombin, meaning transform from fibrinogen, soluble form to fibrin clot. And the MBL is primarily synthesized in the liver, but some small portion expressed in small intestinal mucosal epithelial cells. And I personally believe it should influence on the microbiome. We heard earlier this morning as well as yesterday. And even though MBL is synthesized in the liver, most circulates in the blood and seeps into tissues. So far, MBL has been identified in CSF and alveolar lavage and even in the skin. Now, I'd like to talk about the molecular structure of MBL. So starting from 5', it's so-called system-rich domain, which we think, well, actually it is, to bundle monomer of MBL, and followed by collagen, long collagen lesion. Hence, it is called the collecting. And CRD, carbohydrate recognition domain, which is primary target recognition domain. And MBL has 4 exons, and of which first exon, towards 3' of first exon, there are non-SNP, they're called D, B, or C allele. So if you have any of these, MBL secretion is reduced. And in addition to this, at least 3, actually we know more, 5' open reading region as well as the 3'. And because of these alleles, we have so many haplotypes. This ended up in broad range of plasma MBL levels from undetectable to, we've seen 20 microgram per ml, which is a lot. But average, if you are wild type for both alleles, 2 microgram per ml. And if you have one heterozygous or homozygous for those amino acid substituting mutation, less than 1 microgram per ml. Now, this is the EM. We never published, but just so that I think you can have ideas. So that we usually call it like a bunch of tulips. It's a hexamer, and probably this one's much easier. You can appreciate the 3 of them. So when we started MBL, I've been working on this couple of decades, and only interest was infection. So the first report was the children who have recurrent infection had low or undetectable MBL. And soon afterwards, the life expectancy of cystic fibrosis patients was shorter in MBL deficient people. On the other hand, these MBL deficient people are better against intracellular pathogen. At the time, we knew only 12 TB. So because of this clinical notion, we generated MBL knockout mice. Now I have to mention gene compared to mouse and human. So mouse, they have two protein derived from totally different chromosomes, MBL-A and MBL-C. MBL-A is acute phase protein. So, and while MBL-C is consistently expressed. So when it's acute phase protein, so the naive mice, MBL-A is not really much, but once like LPS challenge, then MBL-A shoots up. Now compared to this, human, we have only one protein, which is genetically homologous to MBL-C. So often some people might describe MBL-C, human MBL-C. But, and if we compare the protein level, actually 40–50% homology between these three human, mouse, A, and C. So when I'll give you phenotypes, I'll talk about MBL knockout phenotypes. So MBL knockout mice are actually double knockout of MBL-A and MBL-C. So we made single knockout and cross to make double knockout, so MBL no mice. So I'd like to mention, so there sounds very similar molecules out there now. First of it, it's LMAN1, which is also called ELG-IC53 or mannose receptor, which we also worked on. But I'm not going to talk anything about this, but both these sound cruelly similar molecules are on membrane protein. And so when we made MBL no mice, mice were healthy and fertile. So this was really great for us. And also we didn't see any obvious phenotype even at the histological level. And we made sure there's no transcript, no protein, and lectin complement pathway was undetectable. Most of the talk I'm going to share are already expressed. So if you would like to see detail, please read the original. Okay, and so first of phenotypes we expected, yes, MBL mice are susceptible to the infection and impaired acute inflammatory responses. So I would like to share two cases. One, Staph aureus blood-borne infection. So we inoculated mice with staph intravenously, and red column for MBL null and gray for wild type. So this data is bacterial titer 2 hours post inoculation. So you can see significant, at least 1 magnitude more bacterial titer in MBL knockout mice compared to wild type mice. And at this 2 hours, we also looked at cytokine TNF alpha IL-6. Both were diminished in MBL null mice. So, and second infection example was a viral infection, influenza A virus infection, and is the primary lung infection. And in this data, MBL null mice are in closed bar and wild type in open bars. So day 1 post infection, still MBL knockout mice lungs could not clear a virus, while wild type almost kind of undetectable. And we also looked at, just part of inflammatory response, we looked at white blood cells and percent of neutrophils. You can see response, these white cell numbers and neutrophil populations in Mbl lungs were different from wild type. And I was drawn to imaging all the time. And at this time, I had only FDG accessible. But I did chronological imaging post-infection day 1, day 4, and day 8. Left panel showing wild type mice and the right panel showing Mbl null mice. So by day 8, there was almost no signal in the wild type mice lungs, but you can see some activity, FDG uptake activity in Mbl null mice. So supporting again, Mbl null mice have aberrant response to inflammatory response. Now, based on the Mbl pathogen, broad pathogen recognition, there is a blood cleansing. It's like a dialysis, but the Mbl cartridge is incorporated. So the idea is to clean pathogen from blood stream. It's early stage of clinical trial. And now from here, I'd like to share with you the phenotype we didn't expect, because we didn't really expect much actually. But surprisingly, first of all, Mbl null mice responded greatly to polysaccharide vaccine. And so we had a collaborator at the time working on group strep type 3 polysaccharide, which was conjugated tetanus toxoid. And altogether, we immunized 3 times, 20 days apart. And this IgG titers are measured at day 60. And this data shows you group strep polysaccharide 3 specific IgG. We also measured just the whole total IgG, IgM. We didn't see a difference between wild type and Mbl. So I'd like you to focus on between wild type and Mbl, and increased IgG isotype was IgG1. And then this increase was in part increased total peritoneal cell number. So peritoneum, that's where we immunized. We used to immunize. And B1 cell was significantly increased in Mbl null mice. And so the next phenotype was coagulopathy. So Mbl null mice had a prolonged bleeding time. And now in mice, you can do kind of simple experiments. So you cut 2 millimeter tip of a tail and put in 37 warm PBS and time till you stop seeing the blood is coming into PBS. So most wild type stops less than 1 minute. While Mbl null mice, many of them didn't stop even 15 minutes. And at this time, we wanted to make sure, the other coagulation factors are not altered in these mice. And as far as we looked at, like platelet numbers and platelet functions were comparable to wild type. And more so when we pretreated these Mbl null mice with recombinant human Mbl, the phenotype went back. So we could say this prolonged bleeding time was due to lack of Mbl. And C3, now by the time we had a mice, people start saying, oh, Mbl is a complement protein, but this result shows you this prolonged bleeding time was C3 independent. Now, as I mentioned earlier, Mbl forms a complex with MASP1. And so we subjected to the same experiment, MASP1 knockout, MASP1 null, and MASP2 null. I'm saying MASP1 null, but actually, so what MASP1 null is MASP1 3 double null, and MASP2 null is MASP2 and SMAP null. So as you can see, MASP1 no mice had prolonged bleeding time, but not MASP2. So we conclude that this prolonged bleeding time coagulopathy was due to lack of MBL and or MASP1. And this work was done by Peter Gall's group. So MASP is directly linked to pro-inflammatory pathway. So here is the MBL's target and MBL MASP complex and PAR, protease activated receptor. There are 4 of them nowadays. And so what happened is when this complex binds to the target, MBL target, MASP can activate PAR and seems to like PAR2. By the way, PAR1 was also known as thrombin receptor. And this PAR activation actually directly linked to pro-inflammatory cytokine signaling pathway. So this is a link between MBL being acute phase and at the molecular level. And the next was bound phenotype. So we used dry bound, so we burned the back of mice. And in the wild type, what happened is ESCA or the burnt skin slough off overnight. But it was not, oh, this is a picture. So it's kind of expensive, but I was told that mice don't really feel pain. Well, any case, so the wild type ESCA shed off, so you can see muscle, back muscle, while MBL no mice didn't. And so most 21 out of 23 wild type lost ESCA like this, but only 3 out of 18. And in other words, 15 out of 18 had kind of looking intact skin. And once again, this phenotype was reversed or corrected by adding pretreating MBL no mice with recombinant human MBL. And again, C3 complement 3 or complement 4, complement 1Q knockout mice were more like a comparable to white type mice. And we wanted to see what's going on. So we did histological analysis and kind of adjacent skin to the burn area. And one of which we did was to look at MMP. We used fluorogenic substrate for collagenase and gelatinase. You can see this green area. Those are MMP active, while we didn't really see much in MBL no mice. And more surprising was MBL no mice, the skin got almost like a 2 or 3 times thicker overnight. And we didn't really do anything further. Now, many of these experiments were done using anywhere between 8 and 12 weeks mice. Now, we wanted to know if there's any age-related issue. So we aged them average like 20 months. And what we saw was nausea, non-alcohol liver steatohepatitis, or simply fatty liver. And when we sent organs to histology, we sent out. And one of the things they noticed was fatty change, meaning the color of liver was actually different. And so we looked at, we did oil red or staining, which stains lipid. So this is wild type and 20 months old. You can see here and there this orange color that means the lipid droplets. In contrast, we saw 2 types of lipid deposit, small deposits all over, or a huge lipid oil red or staining. And we consulted pathology, And they said they see similar pattern in human as well. And we also looked at fatty acid analysis, and 20 out of 23, I think 16 and 18, we had only 3 each, white out and Mbl knockout mice, but 16 and 18 change seems to be higher in Mbl knockout. Now, we never published it. We couldn't find any collaborator nearby. So, but we noticed the Mbl new mice are tame and very sensitive to any stress. I know this characteristic is the most relevant to this meeting, but we haven't really done anything. The one thing maybe close, or at least something happened in the brain is one collaborator at the time was doing traumatic brain injury in the mice. So Mbl new mice had a slow recovery. So, this was just only in vitro. Lastly, this probably relate to brain house cleaning. So this was just like an ELISA type simple binding assay, but amyloid beta 40, 42, and this is just as a kind of control for the experiment. And red crossed bars shows you their binding as 100%. And interestingly, the sulfate sugars inhibited the binding to A beta. And based on this results, even though people didn't really think this system-rich domain as a binding function, it's most likely this system-rich domain, not just bundle, form oligomer, but functioning as a binding domain. And so I'd like to believe that Mbl probably working to maintain balance of amyloid beta. Now we know brain seems to need amyloid beta to brain health, I believe. So I think somehow Mbl participate sort of orchestrating whole innate immune or homeostasis somehow. And so the summary of Mbl effects. So the Mbl status is unnoticeable in general. You wouldn't know, you don't feel it. But it might become an issue if you have underlying conditions and some infections. And it can be 3 ways. Of course, you know, just healthy, nothing. But too little or even too much seems to be not really good. And when I started, I showed you earlier, we had only a few clinical observations, but now the list has been expanding, and autoimmune diseases and also Crohn's disease and some neuronal programs. And also when we started, only deficiency was kind of big deal, but now we know too much is not really good either. Now, I'd like to touch a bit what I'm primarily involved with is the development of PET tracer. To me, seeing is believing, and but pain-free kind of slow development for PET tracer actually. Now, one of which successful is tracer is developed by Dr. Brugaloras, and it's on a phase 1 clinical trial, which is 3F4AP. It's a derivative of MS drug for amylopyridine. So this is a cartoon of a neuron. Neuron is covered or protected by myelin sheath. But if there is any damage, including MS, could be stroke or even Alzheimer, this myelin sheath is damaged and exposed potassium channel, voltage gated potassium channel became exposed, and this tracer can bind. And so this is one, this is, yeah, published. so the, sorry. So we were lucky, I'm sorry, but monkey had an accidental brain trauma injury at this area. And compared to other sort of commonly used tracer, PBR28 or PIB or FDG, CF4AP picked really more strongly. And so the currently under production studies, these tracers are made by radiochemists and metabolic related receptors and some inflammatory related molecules and also pain. And we hope that the tracer, if we hit the right tracer, we could provide early non-invasive and objective diagnostics and possibly therapy too, depends on what we are doing. And lastly, the acknowledgement, I'm member of the Golden Center for Medical Imaging, and particularly thank you to George El-Fakri, he's the director, and my collaborators, Pedro Bugarotas and Norman Dean, and many people are sort of helped me to study Mbl project, and Alain Zuckertz, who cloned Mbl both human and mouse. If I didn't meet him, I would not be working on Mbl, and Mike Super and so forth. Thank you very much.
And thank you, Kazu. We are short on time, not your fault, because you also inherited a little bit of a delay there as well. But what I think we should do is probably move to break until 11:25. So it's a short 7-minute break. I think we all need a little bit time to stretch our legs. But I would encourage everyone to put your questions for Kazu in the Q&A. I know I have a question, which I'll enter into the Q&A. That way, at least we'll both be able to have a break. And